One important aspect of crop productivity is the effect of plant density on crop growth and yield. This is regulated by the process of competition among plants. For reading, we have chapter 12 of the book by Villalobos and Ferreres, Principles of Agronomy for Sustainable Agriculture. Chapter 12 is Plant Density and Competition. Plant population dynamics studies the temporal variation in the number of individuals and their attributes in a population. In the case of crops, these aspects are important to understand the productivity of crops, especially to evaluate the effect of planting density on yield. We need also, or we can use plant population dynamics to understand the wheat crop competition. And third, we need plant population dynamics to understand the process of plant mortality, which may be critical for the establishment of annual crops or for pasture maintenance. Let's analyze what is competition. By definition, a crop is a plant population with individuals with the same genotype or similar genotype and of similar age. Basically, crops are planted at the same date, so their age during the growing season will be very close. The availability of resources that affect crop growth cause competition between neighboring plants. So plants will compete for resources. Higher plants show great plasticity in their growth and in their form in response to the stress imposed by competition. This plasticity is gained by changing the rate of formation, the growth, or the mortality of the organs of the plant. One important aspect in plant population dynamics of plants is that the growth rate of the plant population is proportional to density in the early stages of development. So, the more plants that we have, the greater the crop growth rate. Later, this proportionality is reduced as the competition among plants increases. Finally, we reach a phase when crop growth rate is independent of density and depends only on the amount of resources available. In that period, we talk about an independence of crop growth on plant density, and we name it law of constant final yield. In other words, in the early stages of growth from seed, the biomass of the crop depends on the number of plants present, on plant population. But over time, the supply of resources starts to control the rate of growth of individuals until finally it is the limiting factor of productivity regardless of the density. So we have an initial phase depending on plant population and a final phase where crop growth depends only on resource availability. Any factor that reduces the rate of growth of the plants results in a delayed onset of competition and a reduction in its intensity. The relationship between yield or biomass production and planting density has been analyzed using the so-called reciprocal yield law, which states that the inverse of single plant mass and planting density is a linear function where b1 and b2 are the parameters of this straight line. As the mass per plant is equal to the biomass of the crop divided by the planting density, we can deduce that the yield is equal to harvest index by times planting density divided by B1 plus B2 times planting density, where harvest index is the fraction of biomass that is accumulated in the harvested organ, and dP is planting density. So we have here a relationship between yield and planting density that is hyperbolic. In fact, the meaning of these two parameters, B1 and B2, are related to extreme conditions. When dP is very large, B2 is roughly equal to the inverse of the crop biomass. And when dP is very small, in other words, when we have isolated plants, B1 would be the inverse of the single plant mass. For instance, we have a sunflower cultivar that, when we have isolated plants, can produce 500 grams per plant. On the other hand, the maximum yield is 500 grams per square meter when density is very high. We wonder what is the expected yield if planting density is 5 plants per square meter. Taking the extreme conditions, we can deduce the two parameters of the reciprocal yield law. First, biomass is the inverse of B2 if dP is very high. So B2 is the inverse of 500 grams per square meter. In other words, 0.002 square meters per gram. On the other hand, B1 is the inverse of the single plant mass when dP is very small for isolated plants. Therefore, B1 is equal to 1 over 500, in other words, 0.002 plants per gram. Now, applying the previous equation, we calculate yield as 5 over 0.002 plus 0.002 times 5. In other words, 517 grams per square meter. Another aspect of plant competition is the variability among plants. And that is related to, or that creates a hierarchy among plants. We have smaller plants and larger plants in the field. In this graph, we show for sunflower crops with 10 plants per square meter. This is the distribution of stem diameter. Stem diameter is proportional to plant biomass. So this gives us an idea of the distribution of plant single mass. We plot also the distribution for 2 plants per square meter. So for a very low density, this would be a rather high density, and this would be a low density. We see that for each density we have a large variability in the values of stem diameter. For 10 plants per square meter, we have plants ranging from 8 to 20 millimeters in stem diameter, while for 2 plants per square meter, we have plants ranging from 12 to 20 millimeters in diameter. The question is, what causes this large variability? Or what determines if a plant is small, like here, or large, like here, in a given field? Different studies have shown that the place of an individual in the hierarchy of the population is determined early in the crop cycle. And the amount of biomass produced by an individual in a population under competition is very closely related to the relative order of its appearance, in other words, its emergence in the population. So plants emerging earlier will be larger than plants arriving or emerging later. The advantage of an early appearance is the increased use of resources, because more resources are available if you arrive early, and the deprivation of resources for individuals that appear later. This implies that the likely source of variability of plant mass in the field is variability in time to emergence, which in turn depends on the variation of soil properties, like water content, thermal properties, compaction, or the existence of insects or diseases in the soil, and also on the sowing method. Apart from variability due to variations in the time to emergence, we have another source of variability, which is the loss or the damage to plants caused by herbivores, diseases, or other sources of damage like hail. What is the effect on yield of these plant losses? In this graph we have plotted the yield expressed as percentage of the maximum value as a function of percent of stand reduction. We see two possible functions. The straight line shown here would represent plant losses occurring very late or in large patches. That means that there is no possibility for compensatory growth of the remaining plants. The remaining plants will not be able to fill the gaps created by the loss of plants. Therefore, there will be a straight proportionality between plant density and yield. On the other hand, when we have very early and or random plant losses, the curve would be like this, with a small percentage of plants being lost, there is almost no reduction in yield. So here we have the maximum compensation possible. The remaining plants, after plant losses, will compensate partly for those losses. Another interesting aspect in plant population dynamics is the relationship between density and mortality. There has been a lot of studies on self-elimination, which is density stress induced mortality, that have shown that mortality will occur at high but not at low density. It will start sooner with higher density, and it depends on environmental conditions. Suskachev, in the 20th century, studied the dynamics of self-elimination, in other words, of mortality in populations of spruce close to St. Petersburg. He found that higher densities of trees were found in the poorer soils. He performed an experiment with an annual plant, Matricaria inodora, with either high density or low density, with or without fertilizer. And he observed that the mortality was higher under high fertility. This is plain because high fertility improves plant growth, and that intensifies density stress and provokes more mortality. The mechanisms of plant competition are those that appear as the planting density increases, and therefore the environment of each plant is altered in terms of light intensity and quality and availability of resources, water and nutrients. With higher density, there will be a reduction in radiation interception per plant, in available water and nutrients per plant, and we will have also a change in the far red ratio of light, which is captured by phytochromes and will trigger morphological changes. This will lead to different responses to high density. First, there will be a reduction of expansive growth and mass per plant. We can observe also changes in the harvest index. For instance, with very high planting density, there will be a reduction in harvest index in maize. We will observe a reduction in the number of seeds per plant and or mass of single seeds. We will observe changes in the distribution of dry matter among plant organs. With high density, we will observe an increased allocation to stems and reduced allocation to leaves, and that means an increased ratio height over stem diameter, and that increases the risk of lodging. With high density, we will observe a depressed branching in dicots, for instance in soybean, and reduced tillering in cereals. High density will also cause an acceleration of leaf senescence. It will start sooner and the rate of senescence will be higher, and that seems to occur in response to very low levels of radiation. Finally, we might observe changes in crop quality. For instance, the reduction in size of harvested organ means a reduction in the quality for many horticultural products. The other aspect that was of interest to agronomists is the response of crop yield to plant density. Typically, we can observe two different responses. One is a hyperbolic response, and this is deduced mathematically from the reciprocal yield law, and many times we observe parabolic responses. That would be the case of the response of grain maize to plant density. Here we see an example of the response of total biomass or grain yield of maize to plant density. Total biomass usually will follow asymptotic hyperbolic responses. On the other hand, grain yield might lead to parabolic responses in many situations. This might be related to the existence of other limiting factors for productivity. For instance, when we have conditions of high fertility or low fertility, in general high fertility would show a higher optimum plant density than for low fertility. This is an example for maize where we have a limiting factor like nitrogen for the red curve, and we have also a reduction in harvest index for very high density. The response of yield to plant density depends on the cultivar season length. For instance, in this plot we see that sunflower cultivars, this is based on sunflower data, we have that long season cultivars show this type of response where saturation is achieved at rather low planting densities. On the other hand, short season cultivars like this one would require much higher planting densities to saturate, to reach maximum yield. The response, we said that depends on the cultivar. Here we see a plot of one experiment where different cultivars, differing in growing season length, were compared at different densities. What we can see is that the short cycles reach flowering and there is still a lot of space available. On the other hand, at the same dates, the long cycles have grown more, filling the space, and that means, well, this would be an example for soybean in Florida, where we also see that the long cycles saturate with rather low planting densities, while the short cycles would require much higher planting densities to achieve maximum yield. This is explained because determinate crops stop vegetative growth after flowering. Indeterminate crops do not stop but slows down vegetative growth after the start of flowering. As maximum growth is proportional to time to flowering, because vegetative growth is reduced after flowering, then the long cycle cultivars will keep growing if space is available, in other words, when we have low density, or when we have gaps due to plants killed by pests or diseases. In other words, the long cycle cultivars will use available space, will fill the gaps when needed. Therefore, with lower planting density, we can reach maximum yields, and that doesn't happen with short season cultivars. There is also an interesting effect of the variability in plant density. So we have places in the field where the planting density is higher and areas of the field where planting density is smaller. Well, the idea is that the larger the variability in the density of plants, the larger the decrease in yield. So variability in density of plants is undesirable in agronomic terms. The response will depend strongly on the size of gaps and the possibility for compensation. In other words, if the gaps are not too large and the growing season is large enough, we have the possibility to compensate. So the areas of the field with lower plant density will reach also maximum yield. Another important idea is that the effect of variability should be more pronounced when the response is parabolic, as both lower and higher densities reduce yield below the optimum. In this case, we cannot avoid, if we cannot avoid a very high variability, for instance, if we have problems with the soil or the machinery, we may reduce planting density below the theoretical optimum.